Thank you. I'm going to present today our work about, as she mentioned, about the new tool called FastRecomb for inferring genetic recombination rate in large-scale cohort. But before I jumped into the methodology, I would just have some background and introduction why it's we need a new tool to infer the genetic map. Basically, the background is just if you basically look at the genome and the inheritance pattern, there is always some correlation between two genomic locations that are being inherited together. Basically, the genomes are not being recombined randomly. And by looking at the frequency of those recombinations, we can create a genetic map for the chromosome. So here is just a simple example of the haplotypes and then the offspring that are being produced after the recombination. Each line represents a chromosome or haplotypes. As you can, there are three generations. And you can see, like in the second line, they have the recombination between the blue one and the orange one. And at the end, at the last generation, if you look at that, the gray bars represent the segment that we refer to as identical by descent or IBD segment. By looking at those IBD segments, we can just have the patterns of the recombination rate and estimate what was the recombination frequencies. In the old classical way, that was people didn't have an idea about the genome. They looked at the phenotype data. The most common example was the fruit fly, that people looked at the two phenotypes, the eye color and then the wing characteristic. If the eye color was blue or red, or then the wing was vestigial or normal. Here you can see on the left, that's like a normal wing, and the other on the right is like vestigial, and different eye colors. And let's assume they produce some offsprings. People looked at the number of offspring, and they counted the number from each different types. And they have the parental here on the left, and then you have the recombinants, the mutants. As you can see, the distribution is at equal. They have less recombinant versus the parental. Basically, that number just gives us, by computing the recombination frequency, give us the probability or likelihood that the two phenotype data or two the genomic segments being inherited together. So nowadays, we know that the genome is linear, and then basically, when you talk about the genetic distance, it's usually the recombination rate from the two genomic locations. And in this example here, like from P1 and P2, that's the physical location, physical distance in the units, like base pairs or million base pairs, but the genetic distance might be a lot shorter or bigger based on the recombination frequency. And the unit is usually, basically, people use centimorgans, and each one centimorgans means basically the probability of you have a crossover is 1%. If the distance between P1 and P2 is 100 centimorgans, basically you're probably going to have one crossover between P1 and P2. So basically the goal of the project is just to looking at those the data that we have based on the the old way people can look at the parent offspring and estimate the recombination rate. But usually we don't have all the parent offspring in the data set. We have to look at the population data. From the population data, we want to infer the recombination rates. But it has a lot of application. I mean, if for studying the historical migration patterns, even in medical research, when people do genome-wide association study, usually people just use the centimorgans versus the physical distance, because it's a more reliable measure to see if how two segments are being inherited from a common ancestor. And the issue with that is that each population may have its own unique genetic map. And there are several tools have been developed. One of the older one was the LD-HAT, and the most recent one is IBD-RECOMB. The problem that these tools have is that they are not easily scalable. The older one, LD-HAT, it can barely handle thousands of haplotypes. Basically, the tool was developed for samples if you have hundreds of haplotypes. It's pretty accurate, but basically if you have a lot of data, you cannot take advantage of the data. And it requires some pre-processing. It's very computationally expensive. And there's a more recent tool called IBD-Recomb that is scalable for like thousands, maybe tens of thousands, but it still cannot handle if you have hundreds of thousands of haplotypes or millions of haplotypes. And nowadays, we do have data, basically UK biobank data or some other biobank-scale data that you can find hundreds of thousands or something millions of haplotypes. So the idea is that we can take advantage of those data, just having the more refined genetic map that is population specific as well too. Because I mean, it's not that only we create one genetic map for the humans, because each population, they might have their own unique genetic map, especially at the fine resolution. So before I'm going to talk about the methodology, how we count the recombination rate, just like a simple background about the algorithm that we used here in this work and some other works that our group have been doing. We use a positional Burrows-Wheeler transform. It's a very simple idea. It's just basically, if you have a haplotype panel, and they are all aligned, and each column represents one variant site here, and we got 5, let's say in this simple example, we have 5 haplotypes. If you sort the haplotype panels based on the reverse prefix order, then you can easily find the matching blocks, basically. Here in this one, just being sorted by the last column based on the reverse prefix order, and all the matching segments going to be just like placed adjacent to each other. With the simple basically intuition, we can just start counting the recombination rate. The basic idea is that we avoid counting pairwise IBD segments or pairwise matches, because the pairwise matches could be quadratic in worst case, especially if you have like a very short length in terms of the centimorgans. So the way we calculate that is we split the genome into windows of the same length in terms of physical distance. And then the next step, we find the matching blocks using PBWT. As you can see, as we sort the data, we're going to have matching block here, and we can easily just looking at the matching block and then just counting the recombination event. But there's another caveat. When we're counting the recombination rate, we count the diverging haplotypes. We define a diverging haplotype as the alleles that carry the minor alleles in each block. Within each block, we just have so basically in the first block on top, we just have the red one. In the third column, that and the bottom, we got the 2 on the bottom. And then we look at the divergence value that comes from the PBWT as well, and we update those windows basically, but just adding to the counter basically. Simply just looking at the matching block and then just counting the minor alleles and then just trying to look at their divergence value and update the corresponding window. So after doing that, we calculate the normalized recombination count, and then just looking at the minor allele at each window and using divergence value, we calculate the recombination rate for each window. And then after that, we use the iterations. At each iteration, we just use the calculated genetic map. We probably get a new set of matching blocks, or some haplotypes might just actually added or removed from the blocks, and we calculate again, and each time we're going to have a little modified or changed genetic map here. We did a couple of iterations, and usually it basically converged after like 4 or 5 iterations. We got pretty same result after 5 iterations. So there's another issue with just using PBWT. That was actually one of the main problems. We couldn't use the PBWT directly. Even though it's very efficient, but if you have any mutation, any genotyping errors or recent mutation, we will consider as the recombination basically as a crossover. To deal with that, we developed another algorithm recently to try to smooth the panel by looking at the matching block on the left, the matching block on the right of each variant site, and if we did have the same haplotypes that are matching from both sides, and there was a very few sides, or 1 or 2, that were just like there's a mismatch between those 2 sides, we sort of like smooth the panel. And the runtime for this is linear to basically just, we have like one time where we scan the panel from the left, from the right, and then sort of the correcting the errors. And sometimes the errors we correct are recent mutations. But the goal is just to avoid that those being counted as recombination when we calculate the frequency of recombinations. So now just to a result. We simulated a large panel of 1 million haplotypes using decode genetic map and chromosome 20, and we added different genotyping errors. For the evaluation, we used the correlation coefficient, Pearson correlation coefficient. And the other issue I want to mention is that the focus of our work was to meet region. Basically, we excluded the beginning and the end region of the chromosome, but this is 5 million base pair from the left and 5 million base pair from the right. The one issue is that we didn't have a special treatment for the boundaries. That's going to be a problem that we're going to have to address later on. And then the other thing is that the genetic map we used for decode, we're just missing the first 2 million, 2, 3 million anyways from the beginning. We couldn't actually just evaluate those. So here you can see the result for correlation coefficient using different tools. Here we got FastRecomb here, and LD had an IBD recomb. One point I have to mention that for FastRecomb, we use a lot more haplotypes because of just the pure fact that we could use the chameleon haplotypes to estimate the recombination rate versus the IBD recomb, we used a 5,000 haplotype. And for LD hat, there was only 192 haplotypes that were the pre-computed the lookup table that is required for LD hat. We used that one. And the genotyping error was 0.1% in this, for this experiment. So there's another point with the way we count the recombination and estimate the genetic map. The sample size is a very crucial factor. Basically, if we don't have enough samples, the result may not be very accurate. Here, as you can see here on this top figure, the correlation coefficients are not very strong or very high if we have like only 20,000 haplotypes. As we increase that to 100,000, 200,000, and then we reach the state of the art and then just being slightly better, and then with 1 million, we're going to perform better than other tools. So basically, it's very crucial that we have a lot of sample size when we calculate the recombination rate. And the other thing is the runtime, which is linear to the sample size. Basically, easily scalable. If you have millions of samples, it could be done pretty fast. And then the other issue is that when I talk about the error tolerance, if we don't use the p-smoother, the algorithm to smooth the panel and to get rid of the genotyping errors, the performance would be decreasing a lot. I mean, you can see here, if the genotyping error is high, the correlation coefficients are pretty low here for 0.2%, the error rate, but this is the error rate for the haplotypes. It's basically the genotyping error is like a double than that, it's 0.4%. It is relatively high, but it's still not too extreme, but you can see the performance decreases a lot. Basically, we need to use the p-smoother to smooth the panel. Basically, if the panel doesn't have too many genotyping errors, it should perform well. If it does have a lot of genotyping error, we need to have the p-smoother pre-processing step and then run the algorithm to count the recombination rate. So here's just the runtime and the memory usage of FastRecomb, which heavily relies on the efficiency of PBWT because it's linear to the sample size. We could run like 1 million haplotypes. The CPU runtime is 13 hours, including the time for p-smoother, and then just like counting the recombination rate. I think it does for 5 iterations. And you can see the memory usage is pretty negligible for 77 mega MB. Versus IBD recomb for 5,000 samples, it took 83 CPU hours. We tried to use a larger sample. The job couldn't finish after a couple of weeks. And with LD had only 192. That was the most reasonable number that we could use with the available lookup table that were provided by the tool. So we used our tool and UK biobank data, chromosome 20. And as I mentioned earlier, basically the recombination rate could be population specific, which is apparently. And here just showing that the result for different self-reported ethnic background from UK Biobank. And there's another thing that you can see that I just separated the white British and white, there's an 80-34. It's because the number of Asian British and black British was a lot less than the white British. White British were like 400,000 roughly. So, and the tool just relies heavily on the number of haplotypes basically. But still, you could see that even white British, by using 8,000 haplotypes, it's a lot more similar to the decode that was generated using European populations. So that it just like proved the fact that the recombination rates are indeed in some locations could be population specific. But by just looking at the peaks that we just see here. But it's a very preliminary result. We need to actually to add some other issues and then just create the recombination genetic map for different populations. And more individual might be needed if you want to have for the other Asian British more accurate as the Europeans, as white British here. So just to conclude, the method that I presented, it's very scalable for very large cohorts. Its runtime is linear. And it outperforms other tools if we have a lot of samples, basically. While the other tools may fail to run it without extensive resources, we can use FastRecomb. And the result that they generate could be better if we have, in our simulation, if we have hundreds of thousands of samples. But 1 million haplotypes, we definitely perform better. With 200, 300, it was slightly better than the other tools. So the tools can be downloaded on GitHub, and then there are two issues, there are several issues, but the two main issues that needs to be addressed later. The one is that the treatment of the boundaries, because the boundaries, you're going to have problems with the way we count the IBD segment. It was treated in a way in IBD recount. We're trying to incorporate that methodology in our ways, because it's a little bit different that we count the IBD segment boundaries. But it has to be incorporated in the tools to have an accurate result for the end regions of the chromosome. And the other thing we should agree that the way we calculate the diverging haplotype might create biases. We simply just use the minor allele counts at each block, matching blocks. It could generate some biases, but you tried different methods, even just counting the total number of segments, but the minor alleles basically generated the best result of what you could get. And so with that, I just want to thank the other people who were involved in this project. My previous supervisor, Dr. Shaojie Zhang from the University of Central Florida, Dr. Degui. I joined his team just right after I graduated at UCF. And the other student, William Yu, from University of Texas Health Science Center. And I would like to acknowledge the funding agency, the NIH. And with that, I want to thank you for your attention. And if there's any question, I would like to answer.